Okay, yeah, thanks very much for that invitation. It's a pleasure to speak at this very interesting meeting. And I do think that some of our recent work does fit in quite nicely with the theme. So I'll talk to you today a little bit about some work we've done in formulation of a new type of coatings. And it's quite different than the previous speaker who was essentially making a barrier coating. As I'll show you, in our system, it's actually quite important to have permeability. So it's kind of the opposite. And it also is quite unique, even though we're doing formulation and we're doing this to achieve function, our approach is a bit unusual because we're using bacteria. And as I go along, I'll explain what I mean by metabolically active bacteria, and I'll tell you how we measured it. Okay. I'm just trying to advance, there we go, okay, advance the screen. So maybe just to set the context a little bit, I'll talk to you a little bit about what's known as a biomimetic strategy, which you might have already be familiar with. Essentially, the strategy follows three basic steps. You start by observing a useful property in nature, in an animal or a plant. So an example could be a type of butterfly, such as shown here, which has very pure white wings, and then try to understand where that property comes from. So that usually is related to the structure or the function at the molecular level. And so if you look at the butterfly wing, you can see it consists of these globules, but they're randomly arranged, which would help with the light scattering to give it such a pure white color. And then the third step is to mimic that structure and to try to achieve the same properties as in nature. And sometimes the mimic looks quite different, but the strategy is to use the similar concepts. And if you understand why the structure gives you a certain property, then you can tweak that structure a bit and still achieve the targeted properties. So in this case, this is a group in Cambridge. They used cellulose to make essentially a cellular structure that had these randomly arranged, essentially pockets of air that cause the same light scattering. Okay, so that's just one example, and that was trying to control color. I saw in the program, actually, after I had started preparing this lecture, that Stephanie Berg will be talking, I think at 4:30, and I believe her talk is also describing a biomimetic approach to achieve a white structure. So I won't say more about that, but we could think about other types of properties, such as wetting. So this is an example. You can see the reference here where some scientists looked at the structure of a type of insect called a springtail, and its surface, its skin, is omniphobic, so neither oil or water will wet it. And if you look at it under an electron microscope, you can see different hierarchical structure. So at the larger length scale, you can see these hill-like structures. And then if you zoom in, you can see there's an array of pads. And then if you look under the edges of the pad, you can see there's essentially some sort of nanocavity being formed. And you can see also that the pads overhang a little bit. So it has this curvature underneath the pads. And in this particular reference, which I'm showing you, I think they realized this is a very difficult structure to make in the lab. It's very fine structure. You can see the scale bar of 500 nanometers. So it'd even be quite difficult to print and quite difficult to self-assemble. So in this case, they just took the approach of just making a physical replica in a polymer. And they did start to achieve some of the same omniphobic properties. But you can still try a biomimetic strategy, and I'm aware of some recent work at Loughborough where they used this as an inspiration and got the idea of mixing a variety of sizes to also create a hierarchical structure. And you can see we have smaller particles creating a short length scale structure and then larger particles. And without the overhang, they didn't have the same omniphobic character, but they also saw some very interesting mechanical properties with the polymer reinforcing the silica structures. So you never know, biomimetic strategies can take you, help you discover other interesting properties. So a third example is from a recent project where if you look at the structure of opal, well, first of all, at the macro scale, you see opalescence, you see a variety of colors in natural opal. If you look at that under the optical microscope, you see it's composed of an ordered array, unlike what we saw with the white materials. In this case, it's ordered array of spherical particles made of silica. It's also interesting that most opals contain about 10% water, and consequently also without the silica being fused together, they're usually quite soft materials as well. So lots of people have made synthetic opals. Just looking at it, it does look like what you see in polymer colloid films. And essentially people do take waterborne polymer colloids and make opal structures, but normally you need something with optical contrasts surrounding it. And you need the mono-sized particles to get this close packing. So that was some inspiration for a recent project I've done in collaboration with Isabella Juritz at Surrey. And we started with just a colloidal polymer or latex, put it in a vial. If you dry very slowly, well, if you just let this dry, actually, you start to see the crystallization start at the top. So it's not a sedimentation. It really is still being swept up by the top surface. You let this go, though, you compact the particles together. And what I'm showing you on the left is the resulting opal, but you can see it's a fairly weak opalescence. There's just a bit of pinkish color there. So then we added just a tiny amount of graphene. So in total in the dry film, it's less than about 1%. The wet latex is grayish. If we dry that down, what we see is shown here at two different viewing angles. So the green is when you look top down, and then at an angle view, you can see it looks bluish. And if we look at the structure, it's quite similar to the opal that I showed you previously. You can see this is a cross-section with AFM, and you can see there's close-packed hexagonal array of the particles. And then if we zoom in even further on one of these surfaces, and here I'm showing you an AFM image with some artificial color. So the green is the graphene, and you can see we've added just a tiny amount of graphene. So we're certainly not enveloping the close packed particles. We're just putting a few graphene sheets in between some of the nearest neighbors. And this is quite interesting. As you can see, the structural color is strongly enhanced. And so what's going on here? Well, essentially, just with a very low concentration of graphene sheets, we do get enhanced structural color because graphene has a wide spectral absorbance. So it actually helps to absorb some of the scattered light that isn't the intended color. And also, it has a higher refractive index, and so it adds additional contrast, so to enhance some of the reflectivity at the interfaces. So to show you some of the properties in response to mechanical deformation, I'll just show you this short video. So this is just showing one of the crystals we made by the method I just described. And you can see if you change the viewing angle, you can get different colors. It can go from green to blue. Now we're showing if you apply some pressure, you can see it goes a bit reddish. Essentially, as you change the spacing in periodic structure, you can press on it with your finger, and you can see that also leads to a structural color change. So you don't need a huge pressure, just some light pressure is enough. And finally, under tensile deformation, you can see it also is going from a greenish to a bluish color. So in addition just to having that structural color, it responds to mechanical deformation. Also, I'm not showing data here, but if you look in the reference at the bottom, you'll see it also responds to temperature. Okay, so now back to the theme of adding function by formulating coatings. I'd like to start by asking what sort of coating properties could be desired or might be desired. What would we like to achieve? And let's try to think of some unusual properties. So for instance, could we control color, maybe similar to the chromic coatings I showed you? Could you sense the environment, respond to change? How about, could you have a coating that could remove toxins from the environment? Or maybe that could absorb CO2 or convert it to oxygen? Or maybe that could produce biofuels like hydrogen gas, because that's another big area for things like fuel cells. So maybe a coating could do all sorts of other functions rather than just prevent corrosion or make an aesthetic appearance. And to do all of these things, we're kind of turning the biomimetic idea on its head. So rather than trying to make a coating that copies something as complex as nature, so rather than trying to, because the lists on the left are things that cells can do, but rather than trying to create a cell in the lab, our idea is just to take some cells and put them within the coating. So this is called a biocoating. And as I said, it's the opposite of biomimetic. We're not trying to mimic nature. We're actually just going to use it directly. It's kind of a shortcut. You could think of it that way. So we're adding this bacteria just to waterborne coatings or latex films. So that normally what you do is you start with a stable colloidal dispersion. You cast it on a surface and allow water evaporation. The particles will pack together. And then because of capillary forces and reduction of interfacial energy, those spheres will start to be deformed to fill space. And crucially, and this is important for this project, in the later stages, the polymer chains and neighboring particles will diffuse across the boundaries this by an interdiffusion process. And that will lead to a cohesive or strong film. Because if you don't have that step, then you'll have a much weaker interfaces between the particles and an overall weaker coating. So our approach then is just to take essentially a latex and add to that a living bacteria and to just then make the film. And if you do that, you can see we get a slightly cloudy film, such as shown in the photo. And that's to be expected because you'll get some light scattering between the cells and the surrounding polymer. And I should have said in the previous slide, we're using E. coli in this initial work, just as a model system. So that all looks great, but I guess a key question, and really the theme of the rest of this lecture, is will the bacteria survive the drying process, the dehydration? And then when you have them in a final film, in order for them to be active, they need to be rehydrated. So will it be possible to rehydrate the bacteria when they're in a latex film? So these are questions that we were looking at. And I said it just now that we just add living bacteria to latex. Some of you might have thought, well, that's really weird. Like, wouldn't the latex just kill the bacteria? Well, there is some concern with some surfactants. So SDS is known to be toxic to disrupt cell membranes. We had some very preliminary results that kind of gave us a clue that maybe phosphated anionic surfactants would be less disruptive. So that's what we went with. And we also use non-ionic surfactants, and they're known to be less harmful to cells. And we did with our collaborators some toxicity studies. So here on the left, we're just saying, we're showing that the cell counts over 7 days essentially are stable if you put the cells in just a saline solution. And on the right, in the red bars, we're saying if you just add the cells to a latex, wait 7 days, the cell counts are virtually the same. So there is no sign of toxicity. If you'd like to know more about how the latex is synthesized, I would encourage you to see the poster by Josh Booth, which will be shown later today. Okay, so this is what one of our films looked like. We wanted a durable coating, so we used one with a glass transition of 34 degrees C. And as I showed you in the diagram, we need to get particle deformation and diffusion. So you always have to film form at higher temperatures. So these particular coatings, we chose to film form at 37 degrees C, just in an incubator at that temperature. And that was fine because E. coli can tolerate temperatures up to around 40 degrees C. So that we're looking at the macro scale. What about the microstructure? So if you look at in an SEM at a cross section, you can see where all the arrows are pointing. You can see there are cells that are essentially encapsulated in one of these latex films. And then it's kind of cool that if you zoom in a bit, you can see there's a elongated E. coli cell there. You can see that its surface is a bit dimpled. So what that tells us is the colloids are really pressing up and making good contact with the cells. And from what we can see, the cell isn't harmed by that. It just conforms to its environment. Okay, so I just showed you a picture, an image of the cells in the coating, but what we want is for the cells to do useful things. So to clean the environment, to capture carbon, to do all sorts of other function. And so therefore they have to have a metabolism, they have to be metabolically active. And living cells oxidize a chemical known as NADH, and they oxidize it to NAD+. And if we add to that, if we put a molecule called resveratrol in contact with the cells at the same time, then the resveratrol would be reduced as the NADH is oxidized. And resveratrol is just a bluish molecule, weakly fluorescent. However, when it's reduced, it becomes fluorescent pink. And so we can use the resveratrol as an indicator of the metabolic activity of the cells in the coating. And so you can put the coating just in the bottom of some wells, add the resveratrol and in like a saline solution, and then check the metabolic activity. So on the left, you see some of the wells are blue. That means there's very low or essentially zero metabolic activity. The cells aren't doing anything, they're essentially dormant. On the right, you can see some of the wells are a nice bright pink, which indicates high metabolic activity. But it's not just a yes, no, there can be various levels, and you can see it kind of looks purplish when there's just intermediate levels of metabolic activity. And so by measuring the fluorescence, we can start measuring that. So how did our cells do? Here I'm showing you some examples of one of the earlier experiments. On the left is the metabolic activity we have if we just dry out some E. coli and then rehydrate it. So not in a coating, but just in saline. So we just put them in saline, the medium, and then dry it and then rehydrate. You can see we're getting quite high levels of fluorescence. And to the formulation, we added osmoprotectants. These are things like carbohydrates or sugars, which are known to help in the rehydration and prevention of the desiccation of cells. So on the right, though, we're showing that fluorescence just for the bacteria and a latex coating. And you can see it doesn't look very good. Essentially, there was really no significant metabolic activity. So that was a bit disappointing, but maybe not surprising, because if you have good film formation, then you're going to make a coating with very low permeability. The cells won't be rehydrated, and also they won't be contacted by the resveratrol when they're exposed to the solutions. But this does indicate there's no indication of metabolic activity. So now the formulation comes in. What could we do? So we got this idea of using clay nanotubes to introduce porosity and then therefore increase the permeability. And we're not the first ones to add halloysite. I was actually inspired by the reference down at the bottom here. So they, the group in Clausthal in Germany, used halloysite to add to latex film to prevent cracking. But I also remember them saying, when I saw the paper, that I introduced opacity and porosity. And I thought, if you look on the right, we can see an SEM image. And essentially these clay nanotubes are like matchsticks. And if they're just randomly arranged, you can see all the extra void space that they'll create. So fine, we added some halloysite to the formulation and then looked at the cross section. And essentially this is probably what you'd expect if you know much about formulation. You have the denser halloysite, they're quite large. So in the time of the film formation, they sedimented. And so on the left, we're showing a map of the aluminium. and you can see it's concentrated at the bottom of the film. And then if you zoom in with the SEM, even though this is a mixture of the polymer and the halloysite, you can see that it's very heavily enriched at the bottom of the film by the halloysite. And if you look at the top, it's quite difficult to find any of the halloysite. So clearly we had to solve the problem of the sedimentation, but that was an easy enough thing to do. So we added polyethylene glycol that introduced some steric stabilization for the halloysite. Also started using some sonication to make sure we were dispersing the halloysite quite well. So I'm showing here the original structure of the colloidal film, and then when we add halloysite in film form, and going left to right, you can see those little white components are the halloysite, and at higher concentrations, you can see we have contact or clusters of the halloysite, and you can see just in the cross-section, we've introduced quite a lot of void space between. At the same time, we still have the strength and the cohesion of the polymer binder. So how did these ones do? Well, our first question was, has the halloysite actually significantly modified the permeability? So I have here an example of our permeability cell. On the left, we put in a fluorescent molecule. On the right, just pure water. We put the freestanding biocoating in between the cells, and then we could measure the amount, the rate of fluorescein diffusing through and into the receiver. And to make this quantitative, we created a calibration curve, just intensity versus concentration of the fluorescein. And this is what some example data would look like. Because it takes a certain time for the molecules to get through the thick film, we don't immediately see in the receiver an increase in fluorescence. But after a certain time, then we see this linear increase with the fluorescence in the receiver cell versus time. So that means we're getting this permeability at a constant rate, and we can look at this gradient of how the intensity varies with time, divided by the calibration curve, and from that we can calculate how the concentration of the diffusing dye changes with time. And if we consider the volume and cross-section, in the end we can end up with a permeability coefficient. So I'll tell you more about that in a second. But let me now talk about the mechanical properties. So we could make freestanding biocoatings, essentially make thicker layers in a mold and then lift them out. We could make dog bone specimens for mechanical analysis. So here I'm showing you an example of stress strain curves. We look at our original latex, which has a fairly high strength. You can see it yields at a certain strain. And then when we add halloysite, just say 20%, we get better extensibility, but you can see the strength is compromised and the modulus is actually lower. So even though the clay is a hard particle, essentially because we're getting poorer film formation and more voids being introduced, we actually are diminishing the mechanical properties. And it was very brittle if we added even more halloysite, but if we increase the film formation time, we could at least get a measurable sample. So now I'd like to talk briefly about the trade-off of the properties. So we're aiming for good mechanical properties, but also permeability. So our original latex had a high strength, but essentially no permeability, no measurable, by our test, no measurable permeability. So that was very poor for the biocoating. So great, we could add lots of halloysite to make it highly permeable, but then essentially there was no strength. So a 20% addition was quite good, but to get even higher permeability, we increased the halloysite, but we also increased the film formation time. So that we feel that's really the best compromise. And by increasing the film formation time, we just were able to drive the particle deformation a bit further and encourage some of this interdiffusion, which is so important to get the strength and the cohesion in the coating. So how did this all work? Well, now I'm showing you the fluorescence from the Rizazarin assay. And when we add halloysite, we get much better fluorescence for all the osmoprotectants. And it's similar to what we would see if we just dried it in the medium and rehydrate just with the saline solution. So that really did show that with the added permeability, we could keep increasing the metabolic activity. And we could separately check the viability. So the question is, are the cells actually living? So we had these genetically engineered cells that produced a yellow fluorescent protein, and we could also use propidium iodide, which identified the dead bacteria because it can penetrate the membrane of dead bacteria and stain it. Okay, so with that, I'll summarize, and I hope I showed you that nature can inspire coating design, but rather than mimicking nature, we just use nature or living things directly in a waterborne polyacrylate. It needed permeability, which we achieved for the clay nanotubes, and then we were able to get rehydration after the desiccation of the E. coli. And they were metabolically active from the assays, and they were viable because they expressed the yellow fluorescent protein. And I haven't talked much about the future, but what we're working on now is developing coatings that could help with wastewater treatment, environmental remediation, sensing, carbon capture, etc. And with that, I'd like to thank all of my collaborators. Phil Chen worked a lot with the colloids and the formulation. Simone Kringsb is a microbiologist, PhD student, and the latex was made by the group, Stefan Bonc's group, particularly by Josh Booth, and my collaborator in microbiology is Susie Hingley-Wilsonb. And thank you for your attention.
Joe, super talk. Thank you very much. As somebody says in the comments, awesome talk and interesting ideas. So thank you very much. I'll just pick up a couple of questions from the chat. One question from Justin Perry was, how long do the E. coli live within your halocyte films?
Well, there have been some other people that have made biocoatings with a variety of different species of bacteria. I think if you keep them rehydrated, they can survive for weeks. I think we checked the viability essentially after a couple of days. So I think in future work, we definitely want to look at yeah, the lifetime. But for this particular experiment, we haven't looked more than a few days.
Okay, thanks, Joe. So Whaley asks, in your work on graphene, is there a size effect for the packing of the film and thus the optical properties? And also, does the way you prepare the film affect what happens layer by layer versus rather than mixing, etc.?
Yeah, well, definitely. Well, I think you don't want such big sheets. So the graphene sheets we used were about 350 nanometers, which was just a little bit bigger than the polymer colloids. I do think, at least with nanotubes, if you have really long nanotubes, and I think it would be the same, big sheets could disrupt the packing, and we didn't want that. And definitely in that work, the sample preparation is essential. You have to keep really high order. Well, we didn't do layer by layer, so it's hard to really comment on how it would be different if we had. But this is kind of simpler. It's just a one-step process. Yeah, but it's interesting to note that the crystals form from the top down. So we're not doing a sedimentation. We're just, as it dries, essentially start making a skin layer at the top.
Okay, interesting, thank you. And Bonc Lucas asks, the film formation was done at temperatures slightly above latex TG. Have you looked at the influence of coalescing agents on the bacteria?
Whoa. I guess I'd be a little nervous about adding coalescing agents because it could harm the bacteria. I guess it would be really interesting to find ones that they could use as food or something like that. But the short answer is no, we haven't looked at it. And so the ones as I showed, yeah, we just played with the temperature.